Welcome to the fertilization module, and in this talk we're going to learn about the activation of the fertilized egg in karyogamy in the context of internal fertilization. We will first look at the different types of fertilization, followed by the different types of animals which can be oviparous, oviviviparous, or viviparous. We will then learn about different processes that occur after the fusion of the male and female gametes in the context of mammals. Depending on where fertilization occurs, we have two types of fertilization. In the case of external fertilization, the process of fertilization occurs outside the organism's body. However, in the case of internal fertilization, the process of fertilization occurs within the body of the organism, which is usually a female organism. Thus, in the case of internal fertilization, the sperm must fuse with the egg that is present within the organism. Now, following fertilization, the outcome of that fertilized egg can be different depending on the kind of organism. In the case of oviparous organisms, the fertilized eggs will then be laid out of the body of the organism, and thus further development tends to occur outside the body in the environment. In the case of ovoviviparous organisms, the fertilized egg remains within the body. However, the developing zygote and the embryo will obtain nourishment from the yolk that's present in the egg. In the case of viviparous organisms, the fertilized egg remains within the body of the female. However, the developing zygote and embryo will obtain nourishment directly from the mother by having a connection with the maternal tissues. Regardless of whether an organism is oviparous, ovoviviparous, or viviparous, the egg has to fuse with only one sperm, and thus polyspermy has to be prevented. Now, prevention of polyspermy happens in mammals the way it also happens in sea urchins, except that we don't observe the fast block to polyspermy. So in sea urchins, we observe a fast block to polyspermy by changing the resting membrane potential of the egg cell. But in this case, in mammals, we don't see that fast block phenomenon occurring. However, the slow block to polyspermy is observed where we see cortical granules released into the environment. These cortical granules are present in the egg. So the enzymes present in the egg cortical granules will prevent sperm binding because it has certain components in it, and one such component is ovostasin. Now, ovostasin that is present in the cortical granules is able to cleave ZP2 when it is released during the cortical granule reaction. Cleavage of ZP2 does not allow the sperm to now interact with the egg cell. What also occurs is the protein Juno is released from the oocyte membrane. So the sperm binds to ZP2 and then it uses Juno to be able to internalize into the egg, and by removal of both ZP2 and Juno, polyspermy is prevented, and these events will happen after a sperm has entered into the egg. Now when we look at this picture, we can see that if we have an egg that is expressing a ZP2 mutant which has the inability to be cleaved, we can see multiple sperm that are interacting with the egg. Now, once fertilization occurs and the embryo is in the two-cell stage, we can actually see that there's really not that many sperm or barely any sperm that are interacting with the egg. Now, this particular egg has the normal ZP2 mutant which would have gotten cleaved. When we look at the cell which has the ZP2 mutant that cannot get cleaved, and we observe it at the two-cell stage, we still see plenty of sperm interacting with the cell. And thus, we need the removal of ZP2 to prevent polyspermy. Now let's look at how the fusion of genetic material occurs. Now, once the sperm enters the egg, the sperm chromatin recoils. So if you recall, the DNA that is present in the sperm is very tightly compacted, and hence, once the sperm enters the egg, the sperm chromatin can start uncoiling and get decondensed. Calcium release following fusion activates the proteolysis of cyclin and securin. Now, if you remember from cell biology, securin is a protein that allows mitosis or meiosis to go forward. So securin is the protein that is required to go from metaphase to anaphase. If you remember, in the case of mammals, the oocyte is in a meiosis II metaphase state, so it has not completed meiosis II, and it is in that meiosis II metaphase state when fertilization occurs. Thus, the calcium release following fertilization or fusion allows the proteolysis of securin that will now allow the cell cycle to progress or the meiotic cycle to progress from metaphase to anaphase. Now, DNA synthesis also starts occurring after fusion. However, it happens in the male and the female pronucleus separately, and thus they don't actually fuse with each other immediately. Further, what happens is the centrosome from the sperm will produce a spindle that results in mitosis. So, meiosis has occurred, DNA synthesis occurs in male and female's pronucleus, and then the centrosome from the sperm will produce a spindle that results in mitosis so that we end up with the two-cell stage of the embryo. It is at this two-cell stage that we actually see the diploid nucleus. It should be noted that the sperm provides that pronucleus. It also provides mitochondria, centrosome, enzymes, and RNA during the fusion step. However, it should be noted that the mitochondria get degraded. However, we still will have the nucleus, centrosome, enzymes, and RNA that are present in the egg once the fusion process occurs. Once the fusion process occurs, the mammalian egg cell has to be activated so that embryonic development can occur. This is normally accommodated by a transient rise in cytoplasmic calcium. We had seen in the case of sea urchins that activation required calcium in that scenario as well. However, in that case, there was one wave of calcium that was observed after fertilization. In the case of mammals, there are many calcium waves that will occur that affect the timing or order of events in egg activation. Now, fertilization triggers intracellular calcium release through the production of IP3, the same way that we saw in the case of sea urchins. But in this case, the production of IP3 is actually by a sperm-derived soluble phospholipase C-zeta. Now, the released calcium can have many functions. It can activate the protein symptotagmin that initiates cortical granule fusion. The calcium can also activate calmodulin activated protein kinases that will eliminate translation inhibitors and thus promote embryonic development. We can also have MAP kinases that are activated that allows meiosis to resume. Thus, with this, we come to the end of fertilization in mammals.
With this, we come to the end of our talk, where we learned about the two types of fertilization and what are oviparous, ovoviviparous, and viviparous animals. We further learned about different processes that occur after the fusion of male and female gametes in the context of mammals.